Thank you. I just wanted to mention before I get started that this is definitely not my study. This is a long-term monitoring study that's been conducted by the Palisade Insectary. And a lot of people helped on this study, more than are listed on this slide. But certainly Dan Bean helped to coordinate the whole study. Nina Loudon was hugely important in coordinating everything. Sonia, Karen. Darren Gamot's a statistics professor from CMU. Can you hear me in the back? No. Okay. Thank you. Okay. Darren Gamot's, that's better, right? Darren Gamot's a statistics professor at CMU, ran all of the statistics, and then we had several CMU students who were hired on by the Insectary to do a lot of the field work. And then finally, Eduardo Gonzalez did a lot of the site characterization work for us. And I am here because I know how to use PowerPoint. So I probably don't need to go into too much detail to this crowd on what the northern Tamarisk beetle is and why it was introduced. But needless to say, well, just to go into a little bit of detail of how it works specifically, the beetles scrape away the waxy substances on the leaf cuticles and then go in and damage the mesophyll of the leaves. So it leads to desiccation of the leaves. They turn brown and then eventually drop off the tree. And then multiple defoliations over time lead to carbon starvation in the trees. And they begin a process of canopy, crown, dieback, and eventually can die. We all know that since its original introduction in 2001, that it spread quite rapidly throughout the Colorado, especially the upper Colorado River Basin. This is a new map from the Water Center's first technical report that shows the distribution of the beetle in 2015. But most people, we can probably all agree it's spread faster than a lot of people expected it to. We've seen widespread defoliation events throughout the beetle's range, a lot of observations of mortality. And because tamarisk had become, and still is, one of the most dominant trees along riparian areas in the Southwest, large-scale mortality of this tree is going to bring about major changes in ecosystem structure, function, and composition. So it represents a huge opportunity to study all of those changes that are going on. So the study questions that we had in this study that I'm about to present are, and this is a long-term monitoring project done by the Palisade Insectary. Initially, they focused on the beetles and on tamarisk trees. So they looked at the patterns of diarrhea abundance, defoliation events, crown dieback, and mortality over time. Then I joined the study several years later and was curious about what is happening underneath the tree. So as they die, what's happening to the vegetation. And then as the trees start to die, do we get an accumulation of down dead woody debris, fallen branches underneath the trees? That could have important implications for nutrient cycling. And then finally, what site factors can help predict any of these patterns that we see going on? So most of the data, not all the data, about half the data was collected over a 6-year period, actually 7-year period from 2008 to 2014. So this was done at 10 long-term monitoring sites that were set up by the Palisade Insectary, where they released beetles at each one of the sites, the first one starting in 2005, which was the earliest release date in western Colorado. And there were often multiple releases at each site. It depended on, they needed to get the beetles established. So a variety of releases at each site. So there's 2 sites along the Colorado River, one at Knowles Canyon and one at Horse Thief Bench. And I should also mention, as you'll see, there's a wide variety of sites, some along major rivers and some along very ephemeral canyon drainages. There's one site along the Gunnison River, which you can't see the river from here. It's also along a railroad track. 2 sites along the Dolores River, one near Gateway and one near Bedrock. 2 sites along West Creek. The upper site is actually an ephemeral. At that stage, it's perennial as it gets closer down to the Colorado River. And 2 sites along East Salt Creek. Again, the upper reach is ephemeral and the lower reach is perennial. And then the last site, which is probably the most different than the others, is Flume Canyon, which is, there's an ephemeral drainage draining a canyon in McInnes Canyon's National Conservation Area. So besides there being a variety of this, the size of the rivers, of the drainages that these sites are along, 2 of them are privately owned lands, the 2 sites along East Salt Creek. The rest of them are managed by the Bureau of Land Management. We had 2 sites that were burned before the study started in 2007, and 1 site that was mulched in 2010. There was quite a bit of resprouting of the tamarisk at all of those sites, so they decided to keep those in the study and continue to be monitored. So at each site, so this is one of the sites, there is a single release tree where the beetles were released on, and they permanently marked 50 other tamarisk trees, 12 within a pretty close radius, within around 50 to 100 meters, and then 12 more further out from that. They chose a variety of small, medium, and large trees to try to get a good representation of different sized tamarisk trees. And those were permanently marked, and the same tree was measured throughout the years of the study. Every year, the technicians would come and estimate Diarrhobda abundance of both larvae and adults, so in pretty broad number of classes. The percent desiccation of the leaves, and so this was done by looking at what percentage of the canopy was brown leaves as opposed to green leaves. The percentage of the canopy that was made up of dead branches, and so this is if there's no leaves and they were brittle, they changed color, a variety of factors were used to determine if they were dead or not. And when a tree had 100% of its branches dead, it was considered to be a dead tree. Sometimes, of course, a tree that looks dead one year might spring back to life with a tiny sprig of green the next year. So that did occur, but usually most of the time they were really dead. We measured, to get an idea of crown volume and crown cover, they measured canopy height, length, and width of the green foliage. And so as the green foliage contracted, those measurements decreased. And so all of these measurements were done from 2008 to 2014. For most of the sites, they were done 3 times a season. Some of the sites were difficult to get to, so for 2 of the sites, they were only measured 3 or 4 years. Starting in 2011, we started looking at vegetation, and so beneath, or actually 1 meter away from the trunk of each tree on the east and west sides, we assessed the cover of all species in meter square plots. And then we looked at the cover of down dead woody debris in those plots and divided it into 2 different size classes, less than 1 inch and greater than 1 inch. Those size classes correspond to, in the fire world, fuels that dry out within a day, enough to burn, versus fuels that need more time to dry out and able to burn. In 1 year, 10 soil samples were collected at each site paired with a tree, so we could look at soils related to specific trees. We measured soil salinity, pH, and soil texture. And then finally, Eduardo Gonzalez came out and measured a bunch of site parameters that are related to site hydrology and how likely sites are to flood. So he looked at the river width of the sites, the distance from the middle of the site to the river margin, and the height of the center of the site to the elevation of the river water during the middle of the summer. So looking at some of the results. So I'm going to show you all of the sites, these graphs that I'm going to show you now are all of the sites averaged together, and then I'll show you the variation among the sites. So the abundance, this shows the, this is the abundance of larvae. I'm not going to show you the abundance of the adults over the 7 years. And of course, there's this pattern of, there was a kind of a burst of beetles at the beginning. They went down and then came up again. And the insectary thinks that this is likely, and this will make more sense in a bit, that this is likely because they ate a lot of their food early on in the study, and so the populations declined. The leaf desiccation patterns follow pretty closely the pattern of larval abundance. And finally, the amount of dead branches, the average amount of dead branches found on tree canopies kind of has a steady increase, especially early in the study, and then it levels out for the last several years of the study. But this is, these are averages. There's a huge amount of variation between the sites. The sites are all very, very different. So the real story, I think, here is not these graphs as much. I'm sorry. So with the site factors, we ran a lot of different comparisons to see what site factors were related with different patterns that we were seeing. And I can say we didn't find as many patterns as we thought we would. With bees, canopy dieback, the only things that we found that bees were correlated with was percent sand and river width. So canopy dieback tended to be higher at sites with sandier soil and smaller rivers. Okay, so now to get to the individual site data, these are going to be messy graphs. I tell my students to never show me graphs like this. But so this shows every site as an individual line. So with the abundance of beetles, you can see there's a lot of variation between sites. Early on, there's really just a high number of beetles at a couple sites and hardly any at the other sites. With the brown leaves, it shows the same thing. There's a lot of variation, especially early in the study. There's only really 2 sites that are showing a lot of desiccation. And also there's this annual pattern where the leaves start out green at the beginning of the season, they turn brown, and then they end up green again. This is the percentage of dead branches on the trees, and here, of course, a lot of variation as well. And I want to show you that most of the action is really happening the first several years of the study, and then after 2010, everything kind of remains relatively constant. And so this will show up in a couple other places, so it's important to remember. There's one site I want to point out that really never did much of anything. This is on the Gunnison, and it never really, the beetle never really had a huge impact there at all. This graph here shows Tamarisk mortality, so the percentage of the trees at each site that were considered dead at each year. So again, a lot of variation between the sites. There was mortality, at least up until 2014, at 7 of the 10 sites. So there was no mortality found at 3 of the 10 sites. But this also doesn't tell the whole story, because one of the sites where there was no mortality found was the gateway site. And you could see that there's a lot of things going on here with the beetle. At that particular site, on average, half of the trees had dead branches. So there was still a lot going on there. The site that had the highest mortality up here, which is Flume. Flume Canyon was the ephemeral creek draining a steep canyon. This had the highest mortality overall, 56% mortality. But you can see here's a tamarisk in the middle of all these dead tamarisk that is doing perfectly fine. It hasn't really been touched. So there's a lot of variation within sites, and then, of course, among sites as well. This graph here shows crown volume and cover over time. So this is really a reflection of how it's changing the environment for the plants. And again, you can see that there is a dramatic drop in especially volume, also in canopy area the first couple of years of the study. And then after that, it's either steady or actually it increases a little bit, and this increase is likely due to resprouting of the branches. So canopy volume and cover decreased by 63% and 54% just in the first 2 years of the study. So you can see from this picture, the change really happened pretty quickly, and from there on out, they looked pretty similar. This last picture, you could see a little bit of resprouting. The fallen debris on the ground underneath the tamarisk, these results were not significant, but the larger woody debris, really nothing was going on there. The finer woody debris increased a little bit, but then it went down. So not quite sure what's going on there, but possibly some of it got washed away. But in any case, it's not a dramatic increase. So it looks like that most of the dead branches are staying in the canopy for now, and they haven't really started losing all of those dead branches yet. For the vegetation changes, what we expected, of course, is as you see a reduction in canopy cover and volume, so less transpiring volume, that the plants near here would be happy. They like light, they like water. So we expected overall an increase in vegetation cover underneath the tamarisk. But what we found was no change, really, overall, if you averaged all of the sites. So if you just look at this is the total cover. So it definitely did not increase. It went down a little bit, actually, in 2013. That reduction is mainly due to annual grasses going down in 2013. And I'll get back to why that's probably true in a second. These are all divided up into life-form classes. There's not a single life-form class that has a dramatic increase that we saw. We did do a non-parametric ordination technique, which, to read this, basically we have the life forms in red and the sites in black. So life forms that are located next to a site are more abundant at that site. In general, so there's a lot that we could say about this, but to sum it up, and we also overlaid the site factors. We didn't find that any site factors were related to the patterns of which life forms are found at which site, unfortunately. One thing that's kind of interesting is that the annual forbs and the annual grasses are located very closely together. So they're found sort of in similar abundances across the sites. Not surprisingly, but unfortunately, the two most common species we found were non-native noxious weeds. You could probably guess which the most abundant one was. So that's one of them, yeah. So the most abundant one was cheatgrass, of course. So actually 94% of all annual grass cover was cheatgrass. So that pattern that we saw on the, that pattern that we saw in the other graph of the total cover going down, that was mainly due to cheatgrass going down. I think it has something to do with the fact that 2012 was a really dry year, and so it didn't set a lot of seed for 2013, but that's just a hypothesis. The second most abundant species overall was also a non-native noxious weed, Russian knapweed. So unfortunately, this seems to be a general pattern across all sites. 8 of the 10 sites had more non-native cover than native cover. And of that non-native cover, 73% of it were classified noxious weeds. So regardless of how well the biocontrol agent is doing, there's a lot of secondary weed invasions that need to be taken care of at these sites as restoration progresses. So in summary, so Diarrhobda was definitely effective at controlling tamarisk across these sites, but there was a huge amount of variation across the sites, within the sites, and over time, and the site patterns explained just a little bit of those, but definitely not a lot of the variation. Crown dieback resulted in pretty substantial reductions in tamarisk cover and volume. We expected that to result in a big change in the understory vegetation composition, and we didn't see that happen. The flux of dead wood from dying canopies to the ground apparently happens very slowly. That might be a very good thing. If that happened as fast as canopy dieback, that could create additional restoration problems. A lot of wood material being added to the surface of the soil would probably alter nutrient cycling. And because of the secondary weed infestations, of course, active restoration will be needed at all of these sites, and that could be said with or without biocontrol. All right, and that is. Thank you. So I have a lot of my colleagues here in the room. If I can't answer your questions, I'm sure one of them can.
So you said that after, I can't remember the year, maybe 2013, you're starting to get some resprouting. Would you anticipate that you'd get complete recovery from that eventually?
That's a good question. We've seen so a lot of resprouting at some places, but they never become full, robust trees. But Nina could probably speak to that a bit better.
Even this year, it was pretty stable. It hasn't much at all. So it's less than you would think, given that Tamarisk has basically had a couple of years off from significant defoliation. It really hasn't increased as much as we intuitively think. So I think overall it's still pretty damaged. So we'll just keep track and see how long that lasts. Hopefully it will. The beetles should come back.
Yeah, I think that the picture that showed the canopy volume, the volume stayed pretty low overall, and if it rebounded a lot, you would see it go back up again.
We did see an increase in flowering, more so than an increase in volume. That could be one problem.
Thank you, Mia. Mm-hmm.
If I remember right, you talked about one of the variables that you were measuring was soil salinity.
Yes.
Did you see any difference between those sites that had high salinity concentrations, that is probably around the Paradox Valley area, versus other areas how mortality was affected?
Yeah, that's a good question. So the Gateway site had super high salinity, a lot higher than any of the other sites. And it had, interestingly, it had the highest component of native plant species of all of the sites. Salinity did not come out as a significant factor in predicting mortality or branch dieback or any of those. But there is the biggest component. Probably it had the most native species because it was inland saltgrass was really dominant there. And alkali sacchaton. Any other questions? No.
Thank you.
Thank you.
Appreciate it.